known transcriptional repressor and transcriptional activator domains to allow us to turn on in CRISPR-A or turn off CRISPR-I in a programmable way any gene or any set of genes. In essence, now we had a volume switch where we could control the expression of genes. And this has turned out to be very powerful tools for understanding gene function, as we could, in this systematic and programmable way, explore this. And since this time, there have been really a remarkable array of different effector domains that have been fused onto the CRISPR-Cas systems, probably the most spectacular of which arguably are some of the ones developed by David Liu's lab, where they've used deaminases and reverse transcriptases to create base editing and prime editing, allowing precision changes in the DNA. The other key aspect that made this so powerful, again, was its programmable nature. And that plus other technologies like massively parallel oligosynthesis, the stuff that Twist does, as well as next generation sequencing, has basically created a field of forward genetics in mammalian systems, where you could create a library of guide RNAs, tens or even hundreds of thousands of guide RNAs, each one targeting a different locus in the DNA. So for example, a different gene, and do it in a pooled manner where each cell gets a different guide RNA. So a different gene will be programmed to be turned on or off. And now you can do selective screens, for example, for looking for resistance to a drug like vemurafenib, or looking if, as we were, you're interested in the protein folding in the endoplasmic reticulum, hooking this up to unfolded protein response so that if a gene knocks out, disturbs protein folding in the ER, it would then turn on the UPR, which would turn on a GFP reporter. So you could then fact screen for genes for genes that when you knock down or turn on, modulate function of the ER. In this way, in this really precise way, you can sort of dial in on a specific piece of biology and understand all the genes or all the sets of genes that are impacting that biology. The key aspect of this is you had to define ahead of time which biology you were interested in. And that was great on one hand, but on the other hand, it meant you had to create a new experiment, a new type of screen for every biological problem. So if we really wanted to probe in an unbiased way, in a systematic way, this approach wasn't really well suited, didn't really scale well. So that motivated us in a beautiful collaboration with Yviv Regev's lab to pair these types of CRISPR screens with a complex readout, in this case, single-cell RNA-seq readout. And in these experiments, like the screens I talked about before, we take a pool of guide RNAs. Each cell would get a different guide RNA and therefore have a different gene being perturbed. But now, instead of doing a growth screen or a FACS screen, we would pair this with single-cell RNA-seq so that in each cell, you would read out what the perturbation was, which gene was turned on and off, and what the transcriptional response in that perturbation was. So now you can read out both in this massively parallel way, each effect of each gene and how that effect was pushing the cell in this high-dimensional manifold of its transcriptional response. And this and sort of parallel approaches using imaging-based screens have now allowed us to create systematic genotype phenotype maps that map out the functional consequences of altering any gene. And there are now major efforts, including by the Chan Zuckerberg initiative, their billion cell effort to do this, to create systematic genotype phenotype maps. And we think that these will also be sort of powerful approaches for discovery in the same way the human genome has been a great resource to the community. These types of genotype phenotype maps will be terrific resources, but also will provide the type of training sets or the equivalent of the protein database for protein structure determination that will be great training sets for AI machine learning approaches to create model virtual cells. So all this is terrific from a point of view of discovery, and both for basic mechanisms and for trying to develop identify therapeutic targets. And there's major efforts, some of which I've been involved with, but of course, many beyond my lab are entities I've been involved with, of using CRISPR-based approaches to identify drug targets, including several that are now in the clinic. But CRISPR-I or CRISPR-A as themselves are not particularly good therapeutics. And the reason why is they're transient in nature. So sure, you can turn down or turn up a gene as long as you have the CRISPR-Cas9 expressed in a cell, but as soon as you stop expressing it, the cell goes back to its original state. This motivated us to develop the so-called CRISPR-off approach, in which instead of transiently controlling, repressing, or activating a gene, we would bring in a methyltransferase system that would methylate CpG islands and permanently turn off the expression of a gene. And the reason why this system could have a permanent repression is because we're building off of endogenous systems used by the cell to silence genes, to make sure those genes are never expressed again. And these are used broadly in our genome. But a key application of these, or a reason that these evolved, was to repress endogenous retroviral elements. These are parasitic elements that are hopping into our genome all the time. And the cell has systems for recognizing these and methylating the DNA and making sure that they don't get expressed. And the reason that they're so stable is, in addition to these, the DNMT3A system that is the de novo methyltransferase that puts on the first methyl mark on the CpG element. There's a maintenance methyltransferase, DNMT1. And what this does is when DNA replicates, and of course, DNA replicates by a semi-conservative mechanism, so one strand will be old, one strand will be new. That means one strand's going to have a methyl, another one won't. DNMT1 recognizes these hemimethylated states and then puts in the second methyl on the other strand, allowing them to propagate robustly. And it's the fact that this is an evolved system for maintaining these methylation marks that induce silencing that allows the CRISPR-off to be so potent. And so I give you an example here from the system developed by James Nunes when he was a postdoctoral fellow lab. James now has his own lab at Berkeley, in which he basically appends three components: a CRAB domain, a 3A, which is a methyltransferase, and a 3L domain, which is a helper domain, onto CRISPR-Cas9. And after a lot of engineering to play around with linkers, et cetera, he was able to get a system where you only transiently express the CRISPR-off, in this case for about 10 days, but now got a stable silencing of the targeted and very specific targeting of that single gene. And in kind of a remarkable early experiment, James passaged these cells for 15 months and looked at individual clones, and the vast majority were completely off. And again, this isn't a volume switch. It's not that you partially turn down. When you methylate the gene, it really switches off to a completely undetectable level of expression. And then, so full disclosure, so Luke Gilbert, who developed this approach with me, together with David Liu and Keith Young, started a company, Chroma, now Enchroma Medicine, to try to develop these as a way of silencing genes that were involved in pathology, either toxic genes or genes that contributed to disease. In this case, PCSK9, where the genetics had shown that lowering or even completely eliminating PCSK9 was well tolerated in people, led to a dramatic decrease in LDL cholesterol levels and concurrent decrease in risk for heart disease. So here, and I had nothing to do with these experiments other than advising Chroma, they developed a CRISPR-off targeting PCSK9 delivered it with a lipid nanoparticle to a non-human primate. And what they see is even at really quite low doses, they're able to get silencing across the liver of PCSK9, and this leads to a dramatic 75% decrease in the LDL cholesterol that is robust. It's experiments here and in other studies have now gone out to a year or a year and a half with no evidence of decay. So this is kind of remarkable. A one-time shot, and for the rest of your life, you'll have low cholesterol levels. So this was great. We thought this was a terrific sort of separation of church and state. My lab would be interested in developing new tools and applying this to understand how epigenetic silencing was initiated, how it propagated, and its biological role. And then Chroma and Tune and the other companies in this area would be applying this to develop drugs and therapies, because we didn't know how to develop therapies and didn't have the scale to do this. And all that was fine until I got another visit in my lab, this by this couple, Sonia Valab and Eric Minikal, who were running a lab next to the Whitehead where I am over at the Broad Institute. And I actually knew Sonia and Eric from the time they were interviewing a decade earlier for graduate school. And I knew the remarkable story. So Sonia is a Harvard-trained lawyer. Eric was a city planner. And then that was sort of their nice life plan until the events changed that, which was, sadly, Sonia's mother became sick with a neurodegenerative disease and quickly died. And they subsequently found out that it was a disease called fatal familial insomnia, which is an inherited form of a prion disease. So this is dominantly inherited, and Sonia tested whether she had inherited the disease allele from her mother, and she had. And essentially this meant that with almost complete penetrance, sometime in her 50s or 60s, she was going to have the same fate as her mother. So what Sonia and Eric did, being the people they are, is they dropped what they're doing. They went to graduate school with the goal of developing therapeutic strategies for treating and even more importantly, preventing the prion diseases. And they have a number of different strategies, but they came to our office and said, we have this grant from the NIH that we're applying for. We have to have 3 aims, and the goal of it is to have 3 programs in parallel, and the one that is most advanced will then be funded to have a first in human, so-called IND enabling study. So again, I said, we don't really know much about developing therapeutics, but it was kind of an offer you couldn't say no to and didn't want to. So we've had a wonderful time working with both Sonia and Eric and the spectacular team they put together. And I'll spend the rest of the time talking about those efforts. So first, just a moment about prion diseases. The prion disease is caused by an infectious protein that misfolds into a self-propagating conformation, basically a seed. And what this does then is to take the endogenous protein, which exists in its normal non-pathogenic form, and converts it to the pathogenic form. And that means the pathology, the propagation of the prion infection depends critically on continued expression of the endogenous prion protein, and in particular endogenous prion protein in neurons itself. And in a kind of remarkable experiment from Giovanni Malucci now over 20 years ago, she made a genetically engineered mouse in which they could, using Cre recombinase, silence the prion protein in neurons. What Giovanni found was that you could inoculate with the infectious prion the mice, and then if you turned off, after the mouse was starting to get sick, if you turned off expression, the continued expression from the neurons of the prion protein, it would completely interfere with the propagation of the disease. So what you knew from this is, one, that mice, and now we know several other animals, can live well without any expression of the endogenous prion gene, and that if you can stop expression, the mice would be immune to infection. And in fact, even after they're infected and symptomatic, you can block further propagation of the disease. So unlike so much of human pathology, and especially anything in neurodegeneration, the biology here was completely worked out. We understood exactly what we had to do. Stop expression, particularly in the neurons, of this one protein. So this was really essentially, and this was no longer a science or biology question, it was purely an engineering question. Doesn't mean it's easy, but it means it's a much better defined problem. So the first thing we did was said, can we epigenetically silence the prion protein? In fact, we could. So we could use our CRISPR-off system. And I said, we is really a terrific graduate student, Edwin Newman, and a wonderful postdoctoral fellow, Tessa Bertozzi, who've made a great team on our side of the CHARM efforts. So they transiently transfected in hex cells a CRISPR-off targeting the prion promoter of the prion protein, the gene expressing the prion protein. You get transient expression of CRISPR-off, and then this would allow for permanent silencing of the gene. And this silencing was mediated by methylation of the CPG island upstream of the promoter. And we could show also that this was, again, highly specific. So we were having a little off-target effect on expression of other genes. So now what we had to do is deliver this across the central nervous system. And that's, of course, very challenging. And really the only systems available that an adult can do this are AAVs, in particular AAVs that can cross the blood-brain barrier. Our collaborator, Ben Deverman, had developed a beautiful AAV that could do this in mice, and he's now developed another flavor of this that can do this in humans. And although a mouse brain is a lot smaller than a human brain, the distance between a capillary and a mouse neuron, between a neuron and a capillary in a mouse brain, is about the same in a mouse and a human. So if we can do this in mouse and we can get across the blood-brain barriers, big ifs, there's hope that we could do this in humans as well. The problem is AAVs have a very limited payload, less than 5 kb, and you really wanted even less than that. And Cas9 is a big protein. And when you add on all these other things, so Cas9 itself didn't fit, and maybe you could use these micro Cas9s or Cas systems, but even then it was really trying to shoehorn this in. And this motivated us to go back to what was an older technology, a zinc finger technology for doing targeted DNA binding proteins. And this may seem like a step backwards in a way, but really for epigenetic silencing, zinc fingers had a lot of appealing advantages, not the least of which was their size. So all we had to do was get a zinc finger that bound to the prion gene, and then replace the Cas9 with the zinc finger. All that looked terrific, except we got a kind of a rude surprise, which is when we made the zinc finger version of CRISPR-off, it was able to methylate the target, but also was highly toxic. And so why was this? And it was toxic because of this DNMT3A methylation domain. Why is this toxic? I'll have to jump into a bit about how DNMT3A works in order to explain how we resolve this. DNMT3A is basically exists in an auto-inhibited form. And that makes sense because the cell doesn't want to be methylating CPGs randomly. It wants to do this in a controlled manner. And we had gotten rid of this by taking the methyltransferase domain and taking out these auto-inhibition. So basically we had this really hot methyltransferase that could go in and methylate any place it was brought to. But we also knew that this autoinhibition could be released by binding of a histone tail, thanks to a lot of great foundational work they'd done before us. So this led Edwin to propose that we could use the DNMT3L domain and not overexpress DNMT3A at all, but rather use the fact that 3L can recruit the endogenous DNMT3A. But of course, it would recruit in an inhibited form. So what Edwin proposed is if we could put on this histone tail onto the 3L, we could recruit and activate the DNMT3A. Kind of remarkably, this worked. So he it worked like a charm, so we called it charm for coupled histone tail for autoinhibition release of methyltransferase, but charm's a lot easier to say. And through a rounds of optimization, he was able to show that charm worked, and now we could deliver a safe epigenetic silencer that could readily fit into an AAV, and we could get dramatic brain-wide methylation and silencing of the prion protein. We estimated from sort of knockout studies that about 50% is what you would need to be therapeutically relevant. And we were here at 80, and we've gone in other experiments even higher than that. Then the last twist of this was to put in the self-silencing. I told you the methyltransferase, when it comes in, it methylates and permanently silences gene. So you don't want to continuously express it because continuous expression could only cause problems after that. So what Tessa did was she designed a binding site for our zinc finger charm in its own promoter. So, and then tuned this so that it would first silence the endogenous gene at a slower rate and at a faster rate rather, and then silence itself. And this works, turned out work very well and be highly stable and be critical because when we did or didn't have the self-silencing, and this is a 6-month mouse experiment, if we didn't have the self-silencing, we had nice prion silencing, but we also had a number of off-target effects. And these were dramatically cleaned up. The one gene we see that seems to be changed with the self-silencing is keratin, which must be some sort of contamination, either from the mouse skin or from the experimenter skin here. The rest are below the radar of what you can detect. So we can specifically, robustly, and on at least a scale of 6 months, we've done it out to a year, silence a prion protein. Now with this in hand, we could do the experiment where we inoculate a mouse with a prion, wait till it starts to get sick, and then come in with this AAV as a rescue. And with this, we're able to dramatically extend the life of the mice, even after they're getting sick. And presumably, if you do this before the inoculation, it would, as you would do it for prevention, it would completely stop the infection. So we've now done this, that was for mouse, we've made humanized versions. We have a very specific human targeting zinc finger that targets a human prion. We can silence this nicely in a humanized mouse version and get very robust again, almost a level of minimum level of detection across our humanized mouse. And so we're just about with Sonia and Eric and Ben finalizing the design of the zinc finger charm for the human prion protein. We hope to get the final designs done next month or so, and working with a CRO, Virogen, are starting production of the AAV targeting the human prion using Ben's human blood-brain barrier penetrating AAV. And the aim is to get for the IND either in the second half of 2026 or early 2027. But more broadly, we think that this will be a proof of principle of the ability to target toxic genes across the brain, which could be broadly applicable to diseases that are much more common, including Huntington's and potentially Alzheimer's disease. So with that, I'll end. I think, again, I've mentioned them along the way, but worth repeating. It's been wonderful work from collaboration with Edwin and Tessa. And then none of this would have been possible without the leadership of Sonia and Eric and without Ben's leadership on the AAV. With that, I'll end. Thank you guys for listening and take any questions.
Hi, it's a great talk. Hello. I have 2 questions. So one is when you do this permanent silencing, probably you induce a heterochromatinization as well. How far does the heterochromatin spread?
Yeah, so the view, most CPGs in the genome are methylated. And what you find is that you have these islands of unmethylated CPGs upstream of the promoters about 60% of the genes. And what you see with this, an effective epigenetic silencer, is that it will go in and fill in the methylation on the CPGs, but it won't do it on the genes next to it. And we actually did this genome-wide, both by doing RNA-seq for specific ones, but then we did a genome-wide screen where we targeted every gene with the CRISPR off. And we asked if you were nearby an essential gene. So we showed that if you target an essential gene, you got nice dropout as you expect. But if you're nearby an essential gene, you've even the next gene over, you're no more likely to have an effect than any other place.
So have you measured H3K9 translation or something like that? Is that also restricted?
We have not done a genome-wide H3K9. We've done genome-wide whole genome bisulfite sequencing with these, and that we really see very nicely. It's highly specific.
Yeah. It's very nice. You are trying to attempting to cure the mice with prion disease and so on, but some of the mice still die.
Yeah.
If you measure the brain for the dead mice, do you see actually they were failed for silencing? And
yeah. So why did we not get complete cure? I think there were two reasons. The primary one is just you're talking about a mice that's already starting to get sick. Even if you put in the AAV, it's going to take a few weeks for the expression to go up. And they only have about 2 months. So for people, the difference between the time lag on average between symptomatic and death is about 5 months. For the mouse, it's only about 2 months. About 2 or 3 of those weeks of those 2 months are going to be used for just getting the expression before you can even start to get the silencing. So I think it's a really, really hard challenge, especially for the mice on these short time scale. And what we see with the mice is they just sort of, at the time or a couple weeks after at the time the charm would be starting to have an effect, they're already not eating and not really moving enough. And they, maybe sort of recover a little bit and eat some, but they never go back to being healthy. I think once you have, if you look at, pathology of someone with prion disease, it's really killing, it's a spongiform encephalopathy. It's killing a lot of neurons, and, there's nothing, that damage you're not going to do. You're not going to be able to reverse. So for prion, if you were thinking about it therapeutically, it's much easier for prevention, as is the case with for the third of people with genetic causes of prion diseases. Either that, or you really would want to push the diagnosis so that you can get that very early.
One last question.
On Transplant Institute, my question is, what do you think future editing perspective? Thank you.
Xenotransplants. Well, of course, the gene editing has been used for the xenotransplant because you can take off antigens or endogenous, actually the same sort of endogenous retroviruses that the silencing systems are working. So that's very much reality. Where xenotransplants are going, there must be people. There's certainly people within a stone's throw of here that are much more qualified than me. But of course, it is a very nice application of gene editing.
Okay, please join me in thanking Jonathan for this wonderful talk. Yes. And again, we have a gift for you. Okay. Okay, so let's welcome our third speaker, Dr. Susan Shao. Dr. Susan Shao is a associate professor of cell biology at Harvard Medical School. She received her PhD from the joint NIH and Johns Hopkins program and did her postdoctoral research in the MRC Laboratory of Molecular Biology. So Dr. Shao joined Harvard Medical School cell biology in 2016, and she soon became a leader in the field of protein synthesis and quality control. So her lab explored the molecular mechanisms that monitor protein synthesis and maintain proteostasis, focusing on how ribosome-associated quality control pathways sense translational stalls and triage aberrant lesson chains. So her work has really been recognized by many awards, and most recently, she is selected as an investigator of Howard Hughes Medical Institute. So please join me in welcoming Susan. Stage is yours.
Well, thank you. And I also want to just echo everybody's sentiments. You know, I really appreciate this amazing opportunity to get to know this community a little bit better. And I think it's a really opportune time to meet like this and talk about science. So I'm going to zoom back into molecular, at the molecular level. And my lab, as Bo mentioned, is very interested in understanding how cells deal with nascent proteins in terms of how they get them to the right place and in the right form, and then what happens if something goes wrong during this process. And because I think most of us in this room are practicing scientists, I thought I would try something a little bit different today and then talk instead about a process of how we got through a series of findings rather than just kind of how trying to sell you a story. And so I would like to first go over very quickly a finding that supported a hypothesis, which then motivated work that led to a finding that seemed to support a hypothesis, and then finally where that finding led us. And so the problem that we wanted to address is a problem that has been around in the field of protein localization for some time, which is trying to figure out how cells basically correctly localize proteins to the mitochondria. And my lab is particularly interested in a class of proteins called tail-anchored proteins that have a C-terminal transmembrane domain because ER tail-anchored proteins and mitochondrial tail-anchored proteins have to be delivered to the right membrane post-translationally. And it was unclear how cells are able to differentiate between these two classes of proteins and get them to the right place. And so the approach that we took to try to understand this problem was a biochemical approach. We hypothesized that biosynthetic and quality control factors that deliver these proteins to each of these membranes would directly interact with these substrates. And so what we decided to do was to functionally reconstitute these reactions. So have a model substrate that goes to the mitochondria, and then try to trap the transient interactions that that substrate makes as it's going to mitochondria. And so our substrate is basically has this system that's here. It has an N-terminal tag and a C-terminal transmembrane domain that will have to embed into the outer mitochondrial membrane. The identity of this protein is not important, but it's OMP25. And we can show that the C-terminal transmembrane domain is necessary and sufficient for its mitochondrial localization. And this was work that was done before. But to able to reconstitute this process, we need to do two things. We need to be able to synchronize reactions of the substrate going into mitochondria, and we need a way to keep the hydrophobic transmembrane domain soluble, because otherwise, if it's just in the cytosol, it would aggregate without a chaperone. And so to get around this problem, we did purify this protein in complex with a chaperone called calmodulin. And calmodulin is really nice because it's a calcium-dependent chaperone. And so in the presence of calcium, if we purify calmodulin with our substrate, you'll see that in this fractionation, the substrate co-migrates with calmodulin. But if we remove calcium with a chelator like EGTA, our substrate is released from calmodulin, and in this size fractionation, it becomes heavier even though its molecular weight is smaller because it's aggregating. And so the next thing that we can add to the system is organelles, so mitochondria and ER, that we can purify from various components. And through other assays, we can show that our substrate can insert into the membranes of these organelles correctly. And so finally, the last thing we want to do is to trap these interactors. So we put a site-specific photo cross-linking probe right in the transmembrane domain, or what we think is the localization sequence, so that if we expose these reactions to UV light, it will induce a covalent cross-link with a binding partner. And the nice thing about this is it's very acute, so you can trap things that bind and release potentially. And so as expected, if we do mass spec on the purifications of our substrate, before and after UV, you'll see that with UV light, we get an enrichment of interactors and we get very little background binding on the other side. And so the first protein that we ended up following up on was a protein called the P5A ATPase. It's also known as SPF1 in yeast or ATP13A1 in mammals. And we chose this protein because my Schuldener's lab had already shown, and we can then also show in human cells that it has a phenotype that we would expect, which is that in wild-type cells, our substrate, which is labeled in green, co-localizes with mitochondria, which is labeled in magenta. But if we knock out this P5A ATPase, our substrate, which is in green, no longer co-localizes with mitochondria, so it becomes mislocalized. And then a collaboration in Jan Park's lab at Berkeley, who determined the structure of the yeast SPF1, we can then come up eventually with a hypothesis or a model that the P5A ATPase, which is actually an ER resident transporter, is able to mediate this phenotype, because if mitochondrial proteins become mislocalized to the ER, what it does is it actually uses this ATP-dependent transport activity to kick that protein out of the ER so that it will have additional chances to correctly localize to mitochondria. And so in other work from our lab and other labs that have followed up on this, I'm not going to go into the details, but I just want to emphasize two concepts that I think we've learned from studying ATP13A1 or the P5 ATPase, which is that during this process of biogenesis, mistakes happen quite often. And in our case, these mistakes are often masked by a corrective factor like the P5 ATPase. And the second thing is that the P5 ATPase represents a quality control mechanisms that prioritizes correction over degradation, because only if you remove the P5 ATPase do your mislocalized proteins actually get degraded. So the cell at least tries to fix things before it gets rid of them. So that worked out pretty well for my lab. And I decided, we decided that, okay, this approach seemed pretty good. Let's try it again. And so this led to a finding that seemed to support a hypothesis. And our hypothesis was still the same, which is that biosynthetic factors and quality control factors would bind directly to substrates. And so we did exactly the same experiment again. And the only difference in this cross-linking assay is that instead of using yeast organelles, which is what we did the first time, we used mammalian organelles. And not surprisingly, because we're using a more complex cell type, we got a lot more hits when we were looking at potential interactors. And so to try to narrow down these hits, what we decided to do is that we had done this cross-linking in the presence of everything on ice. We warmed up the reaction, because we know that if we warm up the reaction, our protein is going to insert more fully into the membranes that are present in this reactions. And so then if we do this, where we compare cross-linking partners after warming or before warming, you'll see that there are a number of points labeled here where these are proteins that bind to the substrate and then release the substrate after it's warmed up. And we particularly wanted to focus on these proteins because if you think about a model where there may be a putative targeting factor or chaperone, it would directly bind a substrate in the cytosol post-translationally, deliver it to the target organelle, and then after the substrate inserts into the organelle, that targeting factor would release the substrate. And there's precedence for this type of work when looking at protein targeting at the ER, for example. And so not surprisingly, if we look at the hits of what these are, we get known targeting factors. So examples are SRP54, which is a substrate binding component of the signal recognition particle that targets proteins to the ER. GET3, which is the protein that targets tail anchor proteins to the ER, so the counterpart to our mitochondrial tail anchor proteins, and these ubiquitins factors, which have also previously been known to bind and release mitochondrial membrane proteins in a quality control aspect. And so of these, there were also 4 other proteins that had this behavior. And the first one that we were really interested in was a protein called ARMC1. And we came at this idea with the idea that we're designing an experiment to look for a putative targeting factor for proteins to the mitochondria. And so there are a number of reasons why we thought that this would be a good idea, that ARMC1 is a targeting factor to mitochondria. The first, of course, is that it interacts with our model substrate, and we can recapitulate this interaction directly by cross-linking in a purified system and biochemically assaying this interaction. The second reason is that it looked like a whole day. So this is the AlphaFold model of ARMC1. It has these 2 globular domains and an amphipathic C-terminal domain. And this helical domain here that has an arm motif basically seems to self-chaperone this amphipathic helical domain, which may look like a mitochondrial transmembrane domain. ARMC1 has already been reported to localize the mitochondria. So if we tag ARMC1 with neon green, you can see that it localizes with mitochondria here. And it's also very cool because it doesn't only localize to mitochondria, but it's actually dual localized. So if we do a fractionation, you'll see that it partitions between the cytosol and the membrane fraction, which is also, again, very common for these targeting factors that have to basically shuttle between the cytosol and organelles. And then also excitingly, we knew that ARMC1 has, deleting ARMC1 has a distinct mitochondrial phenotype. So here we're looking at mitochondrial staining, and if you knock out ARMC1, you'll see that the mitochondria basically becomes scrunched and collapses near the nucleus of these proteins. And then lastly, we had evidence that ARMC1 was predicted to interact with an insertase through a study identified by Jonathan Weissman and Rebecca Voorhees' lab that may insert membrane proteins into the outer mitochondrial membrane. And these predictions show that the C-terminal domain of ARMC1 basically interacts with this transmembrane region of MITCH2, which is an interesting modified solucarrier that these solucarriers normally have 6 transmembrane domains, but MITCH2 has 5, and the CTD seems to take the place of the 6th transmembrane domain. And so basically, we had this model that ARMC1 would be a targeting factor, interacts with MITCH2, and then mediate membrane protein insertion at the outer mitochondrial membrane. We can also validate this interaction between ARMC1 and MITCH2 directly. But after more kind of digging and kind of going after this hypothesis, we ultimately decided that this is actually not true for ARMC1. You know, we had a lot of good ideas. We were very excited for some time. And one of the reasons that we ended up concluding that this was not true is because even though ARMC1 interacts with the model substrate, in vitro, we can never really see an impact on substrate insertion by loss of function. And through other assays, we had other evidence that this was actually not what's going on. And so for the remainder of my talk, what I would like to go through is where this finding actually led us and where the biology took us. Okay, so one of the things that we wanted to do, of course, is to take an unbiased approach to try to figure out what ARMC1 was actually doing. And so to do that, we did a proteomics experiment where we made ARMC1 knockout cells and we rescued it with ARMC1. And so not surprisingly, ARMC1 is the main protein that is rescued here. And normally, if you have a targeting factor, a factor that mediates biogenesis, you would expect the substrates to become destabilized if that targeting factor is gone, and then restabilized if it comes back. And so in this case, we did have 3 very specific proteins whose levels and stability depended exclusively on ARMC1. And these were MTFR1, 2, and 1L. And we can validate this biochemically. You can see that if we knock out ARMC1, the levels of all 3 of these proteins decrease. It's rescued if we rescue with ARMC1 and then not with a mutant that can't localize to mitochondria. But these MTFRs aren't mitochondrial membrane proteins. They are mitochondrial fission regulators. They're paralogous but peripheral mitochondrial proteins. And the paralogs basically just have different expression patterns and regulation. And as their name implies, they're implicated in mitochondrial fission, but their actual function is not very clear. And so this had us a link to mitochondrial morphology. And as I showed you before, ARMC1, knocking out ARMC1 also has a distinct impact on both mitochondrial localization or distribution and morphology. And this can be very specifically rescued if we re-express near endogenous levels of ARMC1. And then also it can be highly exaggerated. So if you overexpress ARMC1, you'll see that the mitochondria are no longer localized kind of in the center of the cell, but they get pulled out all the way to the ends of the cells. And this suggests that the levels of ARMC1 are somehow very tightly coordinated with mitochondrial distribution. And so at this point, we were working on the assumption that ARMC1 somehow specifically impacts MTFR stability and mitochondrial distribution rather than the protein biogenesis, which is the route that we took initially. And so downstream analysis, we can purify ARMC1 complexes and show that these are all of the ARMC1 interactors. This is ARMC1 here, and show that the MTFRs interact with ARMC1 quite well. Even though they don't show up here by Kumasi staining very well, it's only because they're far less abundant than ARMC1. And actually, most of the MTFRs that are present in the cell are depleted if you pull on ARMC1. We found that another major interactor are these mirror proteins. And this was interesting because they're the major mitochondrial trafficking adapters that have been studied so far. And so this may link this to the mitochondrial distribution phenotype. And we identified a number of other proteins that I'm not going to go into detail. only to say that because we had such a big milieu of proteins, we decided to try to simplify this problem a little bit more. And we took this elution of these RMC1 affinity purifications and did a further fractionation. And this allowed us to identify that RMC1 partitions between at least two outer mitochondrial membrane protein complexes. The first contains these MTFRs and MIRO, and the second has this protein called DNAJC11, and I'm not showing the data, but this is a complex that also includes MITCH2. And so with these candidates in mind, what we can then do is plug it into AlphaFold, and it gives really nice, actually plausible complexes for how these two complexes can assemble together. And so again, I'm not going to go into details, only to say that RMC1 kind of sneaks through here and also binds through here. It makes non-exclusive contacts with each of the proteins in the complexes, but it's not compatible with the two complexes together. And also, because we can have molecular insights using these AlphaFold models, we can then design mutants that are designed to disrupt RMC1 interaction with one complex or not the other. And I'm just going to call this the orange complex, and this one the teal complex. And so we can validate these mutants. So if we do pull downs with a mutant that should disrupt the orange complex, you'll see that RMC1 no longer interacts well with only the factors in this complex, but it still interacts with the factors in the other complex. And vice versa, if we make the other mutant, you'll see that there's a separation of function. It still interacts with the orange complex, but not with the teal complex. And so using these mutants, we can then, of course, do a little bit of phenotype screening. We can show that not surprisingly, the orange mutant that disrupts this complex fails to stabilize MTFR, whereas the other mutants can still stabilize MTFR. And that's because RMC1 is required for the assembly of MTFR into this complex, and that is what is required for MTFR stability. We can also show that these mutants have distinct impacts on the phenotype of the mitochondrial phenotype. So disrupting the orange complex seems to phenocopy the knocking out of RMC1, and then disrupting this teal complex seems to partially rescue, but not completely, the mitochondrial distribution. And so just as a quick summary, we now think that RMC1 partitions between these two mitochondrial complexes. We think that this is maybe the most important complexes explaining the phenotype that we see because it has a known trafficking adapter in it. And it is also the complex that is required for MTFR stability. And the impact on trafficking, we believe, is because there's a competition between MTFR binding to this MIRO trafficking adapter and a known adapter to dynein and kinesin that binds to the same site on MIRO. And we can prove that this competition exists because if we knock out RMC1, we recover more of this TRAC2 adapter on mitochondrial membranes and less of MTFR1 and 1L. And if we overexpress RMC1, we can compete off TRAC2 and recover MTFR1 associated with mitochondria. And what's really cool, I think, is again, if we knock out RMC1, we get this really drastic collapsed mitochondrial phenotype. And in this knockout background, if we now additionally knock out TRAC2, we can partially rescue this phenotype, suggesting that basically the loss of function of RMC1 is because you have an overactivity of TRAC2 that basically pushes the mitochondria towards the center of the cell. And so again, we think that one of the purposes of this complex is to actually compete with TRAC2, which facilitates retrograde trafficking, and to balance this. And then of course, the remaining question is what about the other complex? And right now, what I can tell you is that we think that it's an active balancer for the abundance of this complex that has a business end on trafficking. And it's a balancer because they're simply partitioning on-off rates of RMC1 between these two complexes. We also think that there's something a little bit more interesting about it, which is that this DNAJC11 protein is an outer mitochondrial membrane protein that interacts with RMC1, and it has a J domain that interacts with HSP70. And very interestingly, if we knock down DNAJC11, you'll see in the membrane fraction that there's less DNAJC11, but paradoxically, there's actually more RMC1. And this doesn't make sense because there's two complexes, and if you take the binding site away from one complex, why would you have more of your protein at the mitochondria? But this can be replicated. So the mutant of RMC1 that fails to interact with DNAJC11 also is more enriched at mitochondria. And so we hypothesize, or we're going with the model that maybe DNAJC11 actually actively facilitates the release of RMC1 from mitochondria. And the way that we can do this is to uncouple this reaction to isolate mitochondrial membranes that contain ARMC1 with or without DNase11, add in more cytosol or buffer, and then assay for release into the supernatant relative to the pellet. And you'll see that if we take wild-type membranes, we get about 50% of ARMC1 coming off, whereas DNase11 is present. If we knock down DNase11, then we have less ARMC1 coming off in this uncoupled reaction. And we can quantify this and show that this is not only dependent on the presence of DNase11, but also dependent on the presence of cytosol and ATP. And then finally, again, that mutant that fails to interact with DNase11 has the same phenotype. So again, it fails to release from the membranes. And we have some preliminary evidence that this may involve that J domain in HSP70 in terms of how ARMC1 is cycling on and off. And so just as a quick summary, again, this took us in a new direction where we thought we were studying a relatively simple model, but it got us to maybe 10 different proteins in 2 complexes. And we think that ARMC1 has a role in balancing the assembly of this complex, which then has impacts on trafficking, and that this is another balancer that helps to regulate the abundance control of this particular complex. So again, the abundance control of the mirror complexes regulates mitochondrial distribution. And we had thought that ARMC1 may be a more general targeting factor, but I still like the idea that it's a bespoke MTFR biosynthetic factor because MTFR can't exist if ARMC1 is not there. And so that explains how if you don't have ARMC1, you have this phenotype because you have more of these TRAC2 complexes that drives retrograde trafficking towards the nucleus. And if you have too much ARMC1, you have persistent MTFR mirror complexes that has the opposite effect and drives the mitochondrial distribution to the other end. And so then I just want to bring back to our, this is one of my top 10 favorite experiments that we've ever done in my lab. And sadly, you can't see it here, but it says unpublished ... forever. I thought it was still a very great idea, but it led us to kind of new territory. And we'll see if it ever does see the light of day, at least in terms of a publication. And with that, I would like to thank everybody who did the work. This work was really driven by Mike McKenna all the way from ATP13A1 to ARMC1. He was helped by Joao. We had a very nice collaboration with Enyang Park's lab and also Wade Harper's lab, and all of our mass spec is done with Joao Paulo and Steve Gigi, and I'm happy to take any questions.
We have time for a couple questions. Okay, so maybe I can get started, Susan. Do you think what kind of peptide sequences we can learn from the ones you've already learned that can localize into the mitochondria, so that you design or engineer, particularly delivering, for example, mitochondria editors into this organelle?
The initial substrates that get into mitochondria or
Yeah.
something else? Yeah, I think that's a really great question. And I think, as Tom mentioned, the sequences that target proteins into mitochondria are pretty well defined, and you can engineer those quite easily. But the sequences that go to the outer mitochondrial membrane, especially, unless they're beta baroproteins, those are really hard to actually identify with high confidence and to be able to engineer. Although once you know one, you can probably put it on, and that's been done before.
Okay. Oh, yeah, we have one more question. Please.
Partially related to track, because you earlier you mentioned this is working with the dining. That part must be over. A lot of track to available. Why it's going to the dining direction instead of anything? Yeah,
no, that's also a great question. And track regulation is still extremely poorly understood. And that's kind of, it's very confusing because there's 2 paralogs of track 1 and track 2 in human cells. And what I didn't show you is that only track 2 has this effect and track 1 does not. But if you just look at AlphaFold or any sequence alignment, you can't really tell what the difference is. And so there's a lot of regulation via post-translational modifications and other aspects that I think are still very poorly understood. People have been working on this, but I don't think there's a very clear answer.
Very nice talk, Susan. I have a question about the MITCH2. So do you have any evidence that ARM C1 is actually doing something with the insertion of proteins?
We tried pretty hard in a biochemical system and in cells, and we didn't see specific activity of ARMC1 in that reaction. So I will say that it's a small proportion of MICH2 that interacts with ARMC1. So what the other MICH2 in the cell is doing is not clear, but our assays were a little bit nebulous in terms of looking at loss of function there.
So you use hello. You use AlphaFold to predict the docking structure of ARMC1, but have you read a paper from Berlin? So they have the protein-wide, not protein-wide, whole mitochondria cross-linking data to see the structural interactions. And have you?
Yeah, so there's beautiful cross-linking mass spec data of mitochondrial proteins, and it really well validates the interaction between ARMC1 and DNA JC11. The MICH2 interaction was a little bit harder, and the reason that we looked at it was twofold. One is because we're already under this hypothesis that it may be a targeting factor, and the second is because in that paper identifying MICH2 as an insertase, ARMC1 is one of the highest impacted proteins. So if you take away MICH2, ARMC1 levels go down.
Okay, thank you, Susan. And again, we have a gift for you. Okay, so let's welcome our last speaker of the session, and also before our lunch. Please join me welcoming Judith Steen. Dr. Steen, she's a associate professor of neurology at Harvard Medical School, and she's also the director of the. Oh, I'm sorry. Dr. Judith Steen is a full professor in the Department of Neurology at Harvard Medical School, and she's also the director of Neuroproteomics Laboratory in Children's Hospital's Kirby Center. Dr. Steen, she's a leader in developing, applying advanced proteomics and systems biology techniques to understand neurodegeneration and regeneration. Her lab innovates high-throughput mass spectrometry workflows and computational pipelines to map proteomes and post-translational modifications in both model systems and human tissues. Their goal is to apply these technologies to identify therapeutic targets in dementia diseases. So please join me in welcoming Judith Steen.
Thank you so much for this wonderful opportunity to speak to all of you here. I'm delighted to be here. So today I'm going to give you a pretty a very big overview picture of what we're doing in our lab and how our field is moving forward. So one of the things that I want to touch on is that we're moving into a different era of proteomics. And these are several of the things that have increased our ability to do some of the experiments that I'll talk about here today. And we've actually, Tienan and Matthias and I have written up a review in Nature that I'd recommend that you take a look at, because we outline a lot of the advances that have happened recently. So my lab is very interested in the question of why we develop protein aggregates in disease. And as we age, all of our tissues and organs will develop aggregates depending on different stresses that you experience over a lifetime. So if you take two individuals, and I want to stress here that the majority of Alzheimer's disease, for example, and neurodegenerative diseases are sporadic. There are no single gene causes for these diseases. So if you have the same risk factors, if you take two people with the same risk factors, perhaps ApoE4, what you'll see is that there'll be different trajectories depending on your life experience. At least this is our hypothesis. And our question is, why do some of you develop disease while others do not? At the age of 85, a third of us will have cognitive decline or full-blown Alzheimer's. So what my lab does is to find ways to identify, understand structure, quantify changes in proteins, and also look at interactions within those proteins and their pathways, as well as post-translation modifications, and look at the kinetics and dynamics over the aging process. So 75% of all aging diseases have tau involved as a pathology. And the question we asked is, why is tau so prevalent? What we do know is that tau oligomers can spread in a prion-like fashion in the disease process. And we basically were interested in defining what that, the version of tau that can spread from cell to cell. And the other thing that motivated us is the current status of tau targeting drugs is that there is no success so far. And so we examined the current disease pipeline. And what we found is that most of them are based on genetic models of single genes. And what happens is these particular models are examined for mechanism, and then we've developed drugs to those model versions of the disease. So the possible reasons for failure at late stages of drug development could be that perhaps the models are not reflective of the majority of disease, which is sporadic. Perhaps we don't have good enough biomarkers to stratify subjects in clinical trials. And also that the biomarkers do not measure target engagement and disease progression. So what we proposed is that we look at large human data sets over the course of disease, understand using machine learning and other deep learning methods now by interfacing clinical data with the molecular data, and find new targets and biomarkers for disease abrogation. And so this is what we've been doing in my lab. And we are developing newer mouse models that reflect the sporadic disease. We are looking at biomarkers that are reflective of disease stage, and also looking for biomarkers that measure the target engagement and disease progression. So this is our strategy that we use. We use mass spec as our central tool for analysis. We look at human brain tissues, post-mortem tissues that are well characterized, but also very well curated by pathologists that we interact with across the country. And we also look at blood samples as well as CSF or body fluids. We then try to understand the tau species by doing very in-depth biochemistry. We do functional studies and develop models and screen for therapeutics for drug discovery. So the central method that we use in our lab was developed in our lab by a former postdoc, Sichen, who developed FlexiQuant, and she is currently a professor at Harvard in the Department of Cardiology and runs her own lab there. And a PhD student of mine, Waltraud, took that forward and developed the FlexiTau analysis method. So what we do is we take endogenous tau from human samples. We prepare or extract the pathologic forms of the protein. We take the digested protein and we spike in a heavy isotope labeled tau with a flex standard, which allows us to do absolute quantification of the protein. And this is completely unmodified, and we measure the unmodified proteins, but the whole platform involves also doing global proteomics and also looking at all possible modifications of the tau. So this is and then we do a directed analysis for the flex peptides. This is a targeted analysis. And we measure the extent of modification of the protein as well as the absolute quant of the protein. So what we find with this is that we can tell which parts of the tau are modified and then do peak tracing and find the modified peptides that are involved in the pathologic forms. We can also tell you what the exact splice variants are, the absolute amounts of the different splice variants in the sample, and we can tell you which parts of the protein are cleaved or modified in any other way. So what does it tell us? It provides us with absolute quantification, stoichiometry, and extent of modification, maps all of the PTMs and cleavages correlated with the extent of modification, and provides us with the patient frequencies of modification. So here's one of the studies that we've recently done, and this is unpublished as yet, but it is in revision and will be submitted shortly. So basically what we can tell is that there are different abundances of the pathologic tau in the different diseases. And this is going to be very key to developing PET reagents or therapeutics, because you have to modify the binding affinity of your drugs and PET ligands in order to accommodate these differences in the absolute amount of the pathologic tau in your proteins. The other aspect is that the specific activity of the ligands that you choose will depend on the target concentrations. And using these, the chemists can actually do the pharmacokinetic and pharmacodynamic modeling with the absolute amounts that we get from these studies. So this is one of the first studies that we did on a large cohort of human subjects with BRAC stages 0 to 6. And it was performed by the people on the slide. And we asked the question, what are the molecular features of the pathologic tau? Which features of the tau differentiate symptomatic from asymptomatic patients? And how or do the molecular features of tau change across the progression of disease? The first thing we found was there were 95 PTMs. That's a lot. So how do you actually look at all of these at once? So what we did was we also looked at the frequency of these modifications in the patients, and we found that some of them were also found in the control subjects, but a lot of them, the disease ones, were found only in disease, but it was not only the usual suspects, meaning the phosphorylations that have been well studied that were important to disease, but there were several others like the ubiquitination and acetylation in the microtubule binding domain. So the next thing we did was we performed unsupervised clustering analysis of the samples, and interestingly enough, the controls fell into one group and a second group. Within the second group, we had higher phosphorylation, but we also had a bunch of patients that were symptomatic as well. What was interesting is that the symptomatic patients also showed this ubiquitination in the microtubule binding domain that the asymptomatic patients did not. Furthermore, as you get more modified tau, you progress from stages 2-3 to 4-5, and in BRAC stage 6, you get massive cleavage of the protein as well, which results in the removal of the C terminus and parts of the N terminus. So tau phosphorylation, while it occurs early in disease and you get an increase in the modification extent in the patients that are progressing further, it does not define the disease as symptomatic. Acetylation and ubiquitination are associated with symptomatic, but not asymptomatic disease. And tau PTM accumulation is ordered and progressive. What does this tell us? This tells us that the pathways that are changing in disease are changing as you move forward. And these are the writer and reader and eraser enzymes that are putting these PTMs and removing them as disease progresses. So the other aspect is the absolute quantification. And one thing I want to draw your eye to is that the different clusters provided a much cleaner separation of the disease and the non-disease patients. And this analysis also revealed the current modification that's used in clinical trials today, the phosphor-217, that is key to differentiating symptomatic from asymptomatic patients. And the other thing that it points out is that the cleavage in the most diseased patients in the Alzheimer's disease have a 7-fold increase in the microtubule binding domain because of the cleavage. And as you can see here, the control, this is N to C terminus. The control N and C terminus are very little modified or cleaved. So why is this important? Because a lot of the biologics so far that were directed as drugs to the tau were targeted to unmodified tau, and they failed. And the other aspect is that some of them were to the unmodified N region of tau. So in order to build these antibody-based drugs, whether it's in cancer or in pathologic neurodegenerative diseases, you have to make sure that your disease form of the protein you're studying has to be characterized really well in order to make an effective drug. So more applications to flex or not to flex your tau. So we did lots of studies on mouse models that are used for Alzheimer's disease preclinical studies. And we found that while you were able to cure mouse Alzheimer's, they do not reflect the late stages of disease. And you can check this publication out. The other aspect that we found is that molecular diversity and clinical progression are dependent on the PTMs of the oligomeric tau that you isolate from the human patients. Patients with faster progression had very specific PTMs associated with their pathologies. And the other aspect that we also looked at was how do PTMs affect structure? So this was a study done on Alzheimer's disease, but also on corticobasal degeneration. And we found that there were very specific PTMs that were influencing the structure of the cryo-EM that we got from the cryo-EM structures. So the tau comparison study was a design of a human cohort with multiple tau pathologies. And in this particular study, we use machine learning and developed different classifiers to classify each disease. We used 2 cohorts of patients from different clinical sites. And in this study, we found that you will likely need very different diagnostic and antibody treatments for the different tauopathies, including chronic traumatic encephalopathy. So what we see is that progressive supranuclear palsy, which happens much earlier in human disease or human aging, is a genetic, it's sometimes genetic, but some of it is also sporadic. It has the most prion-like or most destructive form of tau that progresses the disease much faster than, for example, Alzheimer's disease. And because of that, we think the key to treating the worst forms of tau progression will be to look at the PSP with the most active prion-like activity. And Tien and Micah are now moving forward with these studies and have made cellular models that they can use in drug development for the future. So to conclude that part of the study, one of the things that we're seeing in the global proteomics is that there are many paths to aggregation. And these are druggable paths. And so we have made cell models for some of these studies. And each stress pathway we see leaves an imprint on tau and affects how the disease progresses. And two of our scientists in the lab, Jia, who's a PhD student, and Georg, who's a postdoc, are working on two aspects of the disease that we found for Alzheimer's. One is inflammatory and the other is related to the ApoE four pathways. And we are doing drug screening by modeling those in cell models and then moving on to iPS neurons. So by these studies, we find that we can completely reverse some of these phenotypes that we see in the cell models. And we are also applying this to flexi-syn and synucleinopathies, which we have also studied in a similar fashion. So from this perspective, we are also working on single cell proteomic models to understand how different types of cells are moving forward in terms of disease progression. For example, in PSP, you find astrocytes involved, but in AD, it's mostly neurons. So we want to understand why there are differences there. And interested now with the novel technology that we and others have developed, we can quantify over 2,000 proteins per cell and can find features that are able to define the differences between these cells. We can also do spatial proteomics using FFPE brain tissue, which are abundantly available. And this is using laser capture microdeception. And we are also doing biomarker discovery by building a platform to run high throughput proteomics. And I won't talk more about that, but what I can tell you is that we have moved quite a bit down the pipeline of drug discovery. And I'd like to thank all of the funders and the families that give us tissues or have donated their patient tissues to the brain banks all across the country. And of course, I'd like to thank my amazing team who have been incredible in moving this work forward. Thank you.
We have time for some questions. Please.
Yes.
Great. Thank you. Alzheimer's disease, especially, is not a uniform disease, right?
Yes.
How would you address these PTM signatures to address the patient heterogeneity?
Yes. So that was really key to our including a larger cohort. And we're actually expanding these cohorts even more with Mayo Clinic. And one of the key points that we use to address this heterogeneity is to look at the patient frequencies of the modifications. And the patient frequencies will define which modifications are key to the Alzheimer's disease versus some of the other aspects. For example, we found that there are certain patients who had vascular disease as well as a comorbidity, and they had modifications that were not specific to the Alzheimer's progression, but they were also found on the tau. Yes. 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 So actually, one of the pathways that we're looking at in the ApoE4 patients who do not metabolize fat in an efficient manner, as the ApoE2 or 3, in those patients, there are different pathways that we find dysregulated, and we're looking at how we can recapitulate those. We have recapitulated them in a cell model, and now we're searching for drugs to counteract those.
So I was sort of shocked at the title for one of those papers, because the paper said that tau diversity contributes to clinical heterogeneity. How anyone got away with that title, I have no idea. Contributes suggests causes. All you can say at best is correlates. And the challenge with trying to understand a neurodegeneration this way is the needle in the haystack problem of what's causal and what's correlative. I could even imagine some of these changes are adaptive. And so again, correlation plus plausibility. I can imagine all of this is correct. I can imagine ways that these different forms are pathogenic. But without genetics on your side, you don't really know what's causal and what's correlative.
So, so I think this is really key, because genetics only gives you the possibility of getting disease. And we know that, for example, some of the sporadic diseases are caused by polygenic factors. And yes, that can inform us with respect to how you get disease. For example, the ApoE4 pathways are different from the pathways that you see in the ApoE3 or the sporadic disease. However, one of the things that we see is that the pathways that you see changing in those patients are indicative of whether they progress in disease or not. And this is why we are trying to use new animal models in order to understand those.
Well, I'm happy with there being environmental factors. I'm happy with there being gene-gene interactions. But in the end, and this cuts across all therapeutic areas,
yes,
it's really hard to develop drugs when you don't have genetic evidence that what you're studying is causal as opposed to correlative. And I still think the challenge is, yes, it may correlate perfect one-to-one, who's developing this, that, and the other thing. But again, the question is this cause, effect, epiphenomena? It's just a challenging problem. And so you started throwing animal models under the bus a little bit. But in the end, I think you do have to go back and use genetic tools to see whether some of these modifications, for example, are causal or not.
And this is what we're doing with some of the tau forms. We're using changes or mutations in those tau forms in order to understand whether or not those are the ones that are spreading. The other aspect is that, because the animal models are not always reflective of how humans live. I mean, they live under sterile conditions. They are, for example, with the prion disease, or with tau, you can knock out the tau, and in a normal animal colony, you will have no phenotype. However, we all know that certain animals, for example, the P301L or S are really jumpy. They're hyperexcitable. The A152T has increased neuronal excitability. But unless you stress them, you will not get a phenotype. They are phenotype-less in several publications unless they experience some sort of stressor. And this is where we believe that our work can come in, is we understand the pathway that is induced with the stressor based on the human disease. Of course, we're not saying that this is the cause. And we do state in our conclusions that it is correlative and we have to follow up. And that's why we're developing the new mouse models with respect to the pathways that we observe.
One quick question.
Well, this deep analysis of tau probably, help at least generating hypothesis for searching for causality, I think. So my question is, you show these PTM and splicing so very nicely. However, it's more from the bottom-up approach still. So maybe there are combinatorial relationships between PTM and so on. So is that conceivable to use some reagent to to fish out a certain PTM containing to see what other PTMs may co-occur with that PTM.
So yes, that is absolutely. This is something that we look at as well, is to look at the co-occurrence of certain PTMs at the same time. And you can see this in the studies that we do with larger human cohorts and in the mouse models that we follow up on.
Hey, let's finish the session. And thank you very much, Judith. And we have a gift for you.